Just take a second and think about your body, how incredibly resilient it is. I mean, it's constantly dealing with changes, right? From inside, like nutrient levels going up and down, and outside stuff too, temperature, exercise, stress.
Absolutely.
Yeah.
Yet somehow it maintains this amazing internal stability. It's a concept that really goes back to the 19th century, to Claude Bernard.
Right. He first shed light on it, and
Yeah.
then Walter Cannon, he gave it that catchy name we all know, homeostasis.
Exactly. And, this stability, this homeostasis, it isn't just for the whole body. It drills right down to the cellular level. Every single cell is constantly tweaking its metabolic reaction rates.
Fine-tuning things to keep its own internal balance.
Precisely. And these tweaks happen in response to all sorts of signals. Internally, you've got molecules like 5' AMP or NAD plus DO. They act like little indicators of the cell's energy status.
And externally.
Externally, you have things like hormones, other signaling molecules relaying messages that trigger these metabolic shifts, these adaptations.
So it's really all about balance, isn't it? At every level, from the whole you down to the individual cell. And, well, when that balancing act goes wrong, that's where health issues can crop up.
The sources we're looking at mention several big ones: cancer, diabetes, cystic fibrosis, even Alzheimer's disease. These often involve breakdowns in these regulatory systems.
And it seems like it's often a mix of things causing these disruptions, right? Like genetics, but also environment, diet, lifestyle.
That's right. And sometimes these regulatory pathways get deliberately messed with, even by outside agents. Think about oncogenic viruses.
Oh, yeah. How do they fit in?
Well, some can actually insert their own enzymes, like protein tyrosine kinases, into our cells. They basically hijack the cell's normal signaling for growth control, pushing it towards becoming cancerous.
Wow. So they overwrite the cell's own regulation.
In a way, yes. Or take cholera toxin. That's a classic example. Produced by Vibrio cholerae.
The bacterium causing cholera.
Right. This toxin goes after G proteins linked to an enzyme called adenylyl cyclase in your gut cells. It locks that enzyme in the on position.
So it just keeps making... What does it make?
Cyclic AMP or CCAMP, loads of it continuously. And that causes this massive outflow of water from the cells, leading to the severe dehydration you see in cholera.
That's incredible and devastating.
It is. And one more example, Yersinia pestis, the plague bacterium.
Okay. What's its trick?
It uses a different kind of enzyme, a protein tyrosine phosphatase, to basically dismantle the internal skeleton, the cytoskeleton, of our immune cells, macrophages specifically.
Crippling the immune response.
Exactly. It stops them from engulfing and destroying the bacteria. Just shows how critical these regulatory mechanisms are and how vulnerable they can be.
It really does sound like a biological arms race, and it highlights how finely tuned that internal balance must be. But it's not just external attackers, is it? The material also links things like Alzheimer's and Parkinson's to problems within our own cells.
That's a crucial point. It suggests that malfunctions in the cell's own protein cleanup systems, the ones designed to remove damaged or misfolded proteins, might contribute to the buildup of those problematic protein clumps we see in neurodegenerative diseases.
So our own quality control systems can fail.
They can. And moving beyond just what goes wrong, it's really important to appreciate that these regulatory systems aren't just simple on-off switches.
Right. Not like a light switch.
And not at all. We're dealing with incredibly complex, interconnected control networks. Think less like hitting a single brake or accelerator, and more like a sophisticated system of checks and balances, integrating multiple signals for a really precise outcome.
Okay, so this deep dive is definitely not staying on the surface. Sounds like we're really aiming to get into the nitty-gritty of how our bodies manage this metabolic tightrope walk.
That's the plan.
The outline mentions several mechanisms: passive versus active regulation, controlling enzyme amounts, tuning their activity, this thing called allosteric control,
We'll definitely unpack.
covalent modification, and then these overarching regulatory networks. So our mission really is to thoroughly explore these principles of metabolic regulation using the material here and hopefully come away with a much deeper understanding.
Let's do it.
Okay, let's start with the basics. That distinction between passive and active regulation of metabolite flow, what's the core difference?
Well, passive regulation is quite elegant, really. It mostly comes down to substrate availability, how much stuff is there for the enzyme to work on.
Okay.
And what's really neat is that for many enzymes, their Kelnum value the Michaelis constant.
Which is like a measure of how well the enzyme grabs onto its substrate.
Sort of, yeah. It reflects the substrate concentration where the enzyme works at half speed. And the thing is, This Kallimov value is often pretty close to the typical concentration of that substrate inside the cell. Figure 9:1 illustrates this nicely.
And why is that closeness important?
Because it means that even small changes in how much substrate is around will cause a proportional change in the enzyme speed. If substrate goes up a bit, the enzyme speeds up. If it drops, the enzyme slows down. It's an automatic, built-in responsiveness.
So it adjusts naturally to the supply of ingredients.
Exactly. It gives you this baseline level of coordination, handling the everyday ebb and flow.
Makes sense for internal fluctuations. But the source also says we need active regulation for bigger signals, especially from outside the cell. How does that work?
Right. Passive regulation is great for that internal fine-tuning. But to respond to broader signals, hormones, growth factors, major environmental shifts, the cell needs more direct, often more dramatic, control.
Something more decisive than just substrate levels changing.
Precisely. Active regulation involves specific mechanisms that can quickly and significantly change how well an enzyme works, often overriding that passive response when a bigger, more targeted change is needed.
Okay. Now, something else that stood out was this idea that metabolic pathways tend to flow in one direction in a living cell, even if some individual steps could technically go backwards. Figure 9:2 shows this dynamic steady state idea. Why the one-way street?
That's the concept of the dynamic steady state. Even if metabolite levels seem stable, there's a constant flow through the pathway. Think of it like an assembly line.
Okay.
The product of one enzyme instantly becomes the fuel for the next. So even if one reaction step is near equilibrium, meaning it could theoretically go either way easily.
The next step pulls its product away immediately.
Exactly, pulls the reaction forward. And crucially, the overall energy change for the whole pathway, from start to finish, is usually very favorable, a big negative delta G. That overall downhill energetic slope drives the flow in one direction.
Like the water pipe analogy in Figure 9:3.
Precisely. Even if the pipe has flat bits, as long as the end is lower than the start, water flows downhill. The overall energy drop of the pathway ensures directional flow, despite any near equilibrium steps along the way.
Got it. The overall journey is downhill, energetically. Now let's talk organization. The material really stresses compartmentation, keeping things separate within the cell. Why is that so vital for regulation?
Oh, it's absolutely critical. Physically separating pathways into different locations, organelles like mitochondria, or even just different cytoplasmic zones brings huge benefit.
Like efficiency.
Efficiency, definitely. Take fatty acids. They're built in the cytosol, but broken down in the mitochondria. Separating them prevents what we call futile cycles, wasting energy, building something up, and breaking it down simultaneously in the same place.
Like having separate workshops for construction and demolition.
Exactly. It prevents chaos. Plus, confining unique intermediates to specific compartments lets opposing pathways run at the same time without directly interfering. Glycolysis intermediates in the cytosol are different from citric acid cycle intermediates in the mitochondria. No short-circuiting.
Keeps everything tidy and focused. The source also mentions NAD+ and NADP+ usage as a kind of compartmentation. How does that work? They seem so similar.
They are structurally similar, just a phosphate group difference, and their electrical potential is about the same. But cells use them for very different jobs. It's a functional separation.
Okay.
Generally, NAD+ is the go-to electron acceptor in catabolic reactions, breaking things down for energy, especially feeding electrons into the mitochondrial power generators. It gets reduced to NADH.
And NADP+.
NADP+ is typically used in anabolic reactions building complex molecules. It acts as the electron donor in its reduced form, NADPH.
So different molecules for breaking down versus building up.
Pretty much. It creates distinct redox pools. High NAD plus NADH where energy is extracted, high NADP plus NADPH where biosynthesis happens. It ensures reducing power goes where it's needed. It's a subtle but profound form of organization.
That's really clever. Different currencies for different metabolic jobs. Now, within these pathways, some enzymes seem more important for control than others. Tell us about rate-limiting enzymes.
Right. A rate-limiting enzyme is simply the one that catalyzes the slowest step in a pathway. It's the bottleneck.
Like the slowest car on a single-lane road, dictating the speed for everyone behind it.
Exactly that. Because it sets the pace for the whole pathway, its activity determines the overall flux, the flow-through rate. So naturally, these enzymes are the prime targets for regulation.
Makes sense. Control the bottleneck, control the whole process.
Precisely. Speed it up or slow it down, and you efficiently adjust the pathway's output. The example of statin drugs inhibiting HMG-CoA reductase is perfect.
That's in cholesterol synthesis, right?
Yes, it's a rate-limiting step there. By targeting just that one enzyme, statins significantly lower cholesterol production. It shows the power of regulating these key points.
Okay. So regulating the activity of key enzymes is one thing, but the cell can also control how much enzyme is even there in the first place. Catalytic capacity depends on both enzyme concentration and efficiency. Let's get into controlling enzyme quantity. The source mentions protein turnover, Schonheimer's work.
Ah, yes, Rudolf Schonheimer. His work in the mid-20th century using isotopic labeling was groundbreaking. He showed that proteins aren't static fixtures. They're constantly being made and broken down protein turnover.
Even proteins whose levels seem stable.
Even those. There's a continuous replacement cycle. But for many enzymes, their actual concentration can change dramatically based on signals, hormones, diet, disease states. And these changes come from altering the rates of synthesis and degradation.
So the cell controls both the production line and the disposal system for its enzymes. Let's look at synthesis first. Induction and repression, those sound like gene control mechanisms.
They are, fundamentally. Induction is like ramping up production, often triggered by the enzyme's own substrate, or something similar, an inducer molecule.
How does the inducer work?
It stimulates the gene for that enzyme to be transcribed into messenger RNA, which is then translated into the protein. More mRNA means more enzyme. The classic example is the lac operon in E. coli.
Making lactose-digesting enzymes only when lactose is around.
Exactly. And humans have inducible enzymes too, like tryptophan pyrrolase for breaking down tryptophan when levels are high.
And repression is the opposite, shutting down production when there's too much of something.
Precisely. If a metabolite builds up, it can trigger repression, slowing down the synthesis of the enzymes that make it. Both induction and repression involve specific DNA sequences near the gene cis elements and regulatory proteins transacting factors that bind to them.
Activators turn things on, repressors turn them off.
Basically, yes. The details of how they interact with DNA and transcription machinery are complex. That's Chapter 38 stuff on gene expression. But the key idea is dynamic control over enzyme production based on need. Hormones can also influence this, adding another layer, but we won't dive deep into that now.
Okay, so that handles the enzyme on switch. What about the off switch? Getting rid of existing enzyme molecules. The ubiquitin proteasome pathway comes up here. Sounds complex.
It is intricate, but absolutely vital in eukaryotes for protein degradation. The main player is the 26S proteasome, a big barrel-shaped protein complex. Think of it as the cell's molecular shredder or recycling center.
A shredder? How does it know what to shred?
Good question. Its active sites, the cutting parts, are tucked away inside the barrel, so it doesn't just chew up everything randomly. Proteins need to be specifically targeted.
And that's where ubiquitin comes in, this small tag molecule.
Exactly. Proteins marked for destruction get tagged with ubiquitin molecules. It's a small, highly conserved protein. This tagging, ubiquitination, is done by cascade of enzymes. E1, E2, and crucially, E3 ubiquitin ligases.
E3 ligases. Why are they crucial?
Because there are hundreds of different E3s, and each type recognizes specific target proteins or specific signals on those proteins. They provide the selectivity, attaching ubiquitin often in chains to lysine residues on the doomed protein.
So the ubiquitin chain is like a destroy me label for the proteasome.
That's a good way to put it. The polyubiquitinated protein is recognized by the proteasome, unfolded, fed into the barrel, and chopped into small peptides.
And this isn't just for getting rid of junk proteins.
Not at all. It's essential for regulated turnover of perfectly normal proteins, too. Like cyclins that control the cell cycle. You need to get rid of them at the right time to move forward. It allows for precise, timely changes in protein levels, crucial for countless processes.
The source also mentions how specific these E3 ligases can be, recognizing proteins that have lost a cofactor or are oxidized or misfolded.
Its incredible selectivity. It highlights the pathway's role as a quality control system, not just disposal. It can sense subtle changes. And importantly, the targeting itself can be regulated. Things like phosphorylation, ligand binding, or protein interactions can influence whether a protein gets tagged by an E3 or not.
And we mentioned earlier, if this system gets overwhelmed or malfunctions, it's linked to diseases like Alzheimer's and Parkinson's with those protein aggregates building up.
Absolutely. It underscores just how fundamental this degradation pathway is for cellular health.
Okay, so we've seen how the cell controls enzyme quantity, making them and destroying them. But these are generally slower processes, right? Taking hours sometimes.
Yes, changes involving gene expression and protein turnover take time.
So what about rapid moment-to-moment adjustments? That must involve tweaking the activity of the enzymes already present.
Exactly. That's where regulating catalytic activity comes in. It's much faster.
Yeah.
The two main ways are allosteric regulation and covalent modification.
Let's tackle allosteric regulation first. What are allosteric effectors?
These are small molecules that bind to an enzyme, but not at the active site where the main reaction happens. They bind to a separate, distinct allosteric site.
Allosteric meaning other space.
Precisely. The binding is non-covalent, so it's reversible. And the key is this binding changes the enzyme shape, its conformation.
And that shape change affects...
It can affect lots of things. Its catalytic speed, maybe how tightly it binds its substrate, even where it goes in the cell, or how it interacts with other molecules or responds to other signals.
So it's like a remote control, binding one place to affect behavior elsewhere on the enzyme. And figure 9-4 shows that these effectors often look nothing like the substrate.
Right. That observation led Monod and others to propose the idea of separate allosteric sites. The binding triggers these conformational ripples through the enzyme structure, ultimately altering the active site's shape or chemistry.
Affecting its Kmer, its Vmax, or both.
Exactly. Changing how well it binds substrate, K-series effect, or how fast it works at maximum capacity, V-series effect, or a combination.
The source groups these effectors into 3 types: feedback effectors, indicator metabolites, and second messengers. Let's start with feedback. That sounds intuitive.
It is. Feedback vectors are typically end products of a metabolic pathway that regulate an enzyme within that same pathway. Often the end product inhibits one of the first committed steps.
Preventing buildup of the final product.
Exactly. Classic feedback inhibition. Imagine A goes to B, goes to C, goes to D. If D builds up, it might bind to the enzyme converting A to B and slow it down.
Like the serine example in figure 9-4. Serine inhibiting the first enzyme in its own synthesis pathway.
Perfect example. Serine, the end product, inhibits 3-phosphoglycerate dehydrogenase, the enzyme catalyzing the first unique step. If serine levels get high, it puts the brakes on making more. It's usually a localized control mechanism within that specific pathway.
Makes perfect sense. Self-regulation. Now, what about indicator metabolites? How are they different?
They're also metabolic intermediates or end products acting as effectors, but their scope is broader. They act as signals about the cell's overall metabolic state, coordinating flow across multiple interconnected pathways.
So they have a wider influence than feedback effectors.
Yes. For example, citrate. It's part of the citric acid cycle, central energy production. But high citrate levels also activate acetyl-CoA carboxylase, the first step in making fatty acids.
Why does that make sense?
Well, high citrate suggests the cell has plenty of energy and building blocks from the cycle. So it signals, okay, we can afford to store some energy as fat now. Activate fatty acid synthesis.
Ah, connecting energy production to energy storage.
Right. Or, conversely, alanine, an amino acid made from pyruvate, glycolysis end product, can inhibit pyruvate kinase, a key glycolysis enzyme. It helps balance glucose breakdown versus amino acid metabolism.
So they're like metabolic status reporters coordinating different branches of metabolism. Very cool. And the third type, second messengers. These sound linked to outside signals.
Exactly. Second messengers are specialized allosteric ligands made or released inside the cell in response to an external signal. A first messenger, like a hormone binding to a receptor, or maybe a nerve impulse.
They relay the message inward.
Precisely. They bind to and regulate downstream targets, often enzymes. Cyclic AMP, CAMP is a classic, made by adenyl cyclase when epinephrine binds its receptor. CMP then activates other things like protein kinase A.
Calcium ions, too.
Calcium Ca2+ is huge. Nerve impulses or hormones can trigger its release from internal stores, like the ER. The flood of calcium activates calcium-sensitive proteins, including enzymes for muscle contraction or glycogen breakdown, like phosphorylase kinase.
What else?
Others mentioned are cyclic GMP, CGMP, nitric oxide, NO, polyphosphonacetals. They allow rapid, often amplified internal responses to external cues. Chapters 18, 42, 51 go deeper, but the main point is they link outside signals to inside allosteric regulation.
Okay, that covers the types of allosteric effectors. Now, the source highlights aspartate transcarbamylase ATCase as a great model enzyme for this. Why ATCase?
ATCase is fantastic because its regulation is so well understood. It catalyzes the first committed step in making pyrimidine nucleotides building blocks for RNA and DNA.
Okay, and how is it regulated?
It shows classic feedback inhibition. CTP, a pyrimidine end product, inhibits ATCase, stops the cell making too many pyrimidines.
Makes sense.
But what's really interesting is it's also activated by ATP, which is a purine nucleotide.
Ah, so it senses both types of nucleotide bases.
Yes. This provides a beautiful way to balance the supply of purines and pyrimidines. When ATP is high, signaling lots of energy and maybe the need for DNA replication.
The ATP activates ATCase, boosting pyrimidine production to match the purines.
Exactly. It overrides the CTP inhibition. Conversely, high CTP inhibits it. It ensures the cell keeps the right ratio of building blocks, especially when gearing up for growth.
That's incredibly elegant coordination. Okay, let's shift gears to the other major way enzyme activity is rapidly controlled: covalent modifications. Forming or breaking chemical bonds with the enzyme. These can be irreversible or reversible. Let's start with irreversible partial proteolysis.
Right. Irreversible here means limited, specific cutting of peptide bonds within an inactive precursor protein, a proenzyme or zymogen. Once cut, it can't be easily undone in the cell. The activated protein stays active until it's degraded later.
A one-way activation switch. The example given is chymotrypsin activation. It starts as inactive chymotrypsinogen. How does it get switched on?
Chymotrypsinogen is a classic zymogen. Activation involves cutting specific peptide bonds. First, another enzyme, trypsin, which itself needs activation, makes an initial cut, forming an intermediate pichymotrypsin.
Okay.
Then this intermediate acts on itself and other chymotrypsinogen molecules, snipping off a couple of small peptides. This results in the mature, fully active alpha-chymotrypsin.
What does the cutting actually achieve?
It triggers crucial conformational changes. It's like releasing tension in the folded protein. These changes bring the key amino acids needed for catalysis, His57, Asp102, and Ser195 together in just the right 3D arrangement to form the working active site. Figure 9-5 shows this convergence.
So cutting specific bonds allows the active site to assemble correctly. The source notes many digestive enzymes in the pancreas are stored as zymogens. Why there specifically?
It's pure self-preservation for the pancreas. These enzymes are designed to break down proteins and fats if they were active inside the pancreatic cells that make and store them.
They digest the pancreas itself, leading to pancreatitis.
Exactly. Storing them as inactive zymogens keeps them safe until they're secreted into the small intestine, where digestion is supposed to happen.
And how are they activated in the intestine?
There's an enzyme called enteropeptidase in the intestinal lining. It specifically activates trypsinogen to trypsin. Then trypsin acts like a master switch, activating all the other pancreatic zymogens, chymotrypsinogen, proelastase, etc. It's a cascade.
A rapid, coordinated activation right where it's needed. Pepsin in the stomach is a bit different.
Yeah, pepsinogen activates itself. The highly acidic stomach environment triggers it to auto-catalyze its own cleavage into active pepsin. But the principle of activating only in the right place is the same. And yes, premature activation in the pancreas is the cause of pancreatitis.
A very neat safety and activation system. The source also links the zymogen activation idea to blood clotting and the complement system in immunity. Why is it important there?
Speed and localization are paramount in those systems. You need a very rapid response to wounding or infection, but only exactly where it's happening.
Right. You don't want blood clotting everywhere or the immune system attacking healthy tissue.
Precisely. So the components of these cascades circulate as inactive zymogens. A local trigger tissue damage for clotting, bacterial surfaces for complement, starts a chain reaction.
A cascade, like with digestion.
Exactly. Each activated enzyme activates the next zymogen in line. This amplifies the signal incredibly quickly and keeps the response focused at the site of injury or infection. It's like biochemical dominoes. Preposition for a fast, localized response. Chapters 52 and 55 have the details.
Okay, so irreversible proteolysis is like setting off a prearranged rapid-fire activation sequence. Now for the reversible covalent modifications: acetylation, ADP ribosylation, simoylation, and the big one, phosphorylation. Reversible means the original protein can be restored, but maybe not by simply running the modification reaction backward.
That's the key distinction, yes. The protein gets back to its original state, but the removal often uses a different enzyme and reaction than the addition, usually for thermodynamic reasons.
Making the addition might be easy energetically, but reversing that exact step might be hard.
Often, yes. So the cell uses a separate enzyme for removal, frequently a hydrolysis reaction, which is itself energetically favorable.
Let's use phosphorylation as the main example. A kinase adds a phosphate from ATP, and a phosphatase removes it.
Exactly. Protein kinases transfer that terminal phosphate from ATP onto a hydroxyl group of a serine, threonine, or tyrosine residue on the target protein. That's energetically favorable because you break ATP's high-energy bond.
And removal.
Protein phosphatases catalyze hydrolysis using water to break the phosphate ester bond, releasing inorganic phosphate and regenerating the hydroxyl group. It's a separate reaction, not just kinase in reverse.
Okay. And similarly for acetylation, an SC transferase uses acetyl-CoA to add the acetyl group.
Very correct. Acetyl-CoA is the high-energy donor, not NAD+, as the text briefly misstates for this addition step.
Right. And then deacetylases remove it. Some just hydrolyze it off.
Yes, the HDACs, histone deacetylases, do that.
Yeah.
But then you have the sirtuins, another class of deacetylases, which actually use NAD+ as a co-substrate, producing nicotinamide and O-acetyl ADP ribose alongside the deacetylated protein. Different chemistry for removal.
Got it. So reversible means the state is reversible, not necessarily the reaction path. Now, these modifications, especially phosphorylation and acetylation, regulate tons of key proteins, including histones. Let's talk about the histone code. Sounds intriguing.
It is. Histones are the proteins DNA wraps around to form chromatin in our chromosomes. They get decorated with a whole array of chemical modifications: acetylation, methylation, phosphorylation, simulation, ADP ribosylation.
A lot of different tags.
A huge right. And these tags don't change the DNA sequence itself, but they profoundly change how the chromatin is structured and how accessible the DNA is.
Affecting which genes get turned on or off.
Exactly. Some combinations of marks lead to open, active chromatin, letting genes be transcribed or DNA replicated. Other combinations lead to tightly packed, silent chromatin. The specific pattern of marks acts like a code read by other cellular machinery.
And this is epigenetics. Heritable changes without changing DNA sequence.
Precisely. The histone modification patterns can even be passed down to daughter cells, influencing their gene expression. It's a layer of information on top of the genetic code.
Amazing, like annotations written on the DNA packaging. Now, phosphorylation, why is it such a common regulatory switch?
Several reasons make it so versatile. First, there's a huge number of kinases and phosphatases in the cell, allowing for very specific control over many targets.
Specificity and scale.
Right. Second, it's fast and easily reversible. Kinases switch it on, phosphatases switch it off, allowing rapid responses. Third, the phosphate group itself have useful properties.
Like what?
It's got a strong negative charge, which can drastically alter electrostatic interactions, cause shape changes, form new salt bridges, or stabilize structures through hydrogen bonding. Even when added far from the active site, it can trigger conformational changes that affect activity, like an allosteric effect.
And phosphorylation doesn't always mean activation, right? Table 9:1 shows it can inhibit some enzymes and activate others.
Absolutely. It depends entirely on the specific enzyme and where the phosphate is added. Acetyl-CoA carboxylase and glycogen synthase get inactivated by phosphorylation. Glycogen phosphorylase and its kinase get activated.
So it's context dependent, and the effects go beyond just activity.
Oh yes. Phosphorylation can change where a protein goes, how quickly it's degraded, how it responds to allosteric effectors, or even if it can get other modifications.
And proteins can have multiple phosphorylation sites, targeted by different kinases responding to different signals.
Yes. This allows for incredible integration of information. The protein's overall phosphorylation status reflects the sum of many different input signals. It becomes a kind of signal integrator.
Processing multiple inputs to decide on an output.
Exactly. Plus, some kinases phosphorylate multiple targets, coordinating entire pathways, like AMPK, which responds to low energy by phosphorylating and inhibiting both cholesterol and fatty acid synthesis enzymes simultaneously.
Shutting down energy-consuming building projects when energy is low?
Right. So you get this complex interplay. Kinases, phosphatases, multiple sites on one protein, allosteric sites, other modifications. It forms these sophisticated regulatory networks, integrating tons of information.
Okay, so we have these fast reversible mechanisms, allosteric and covalent modifications like phosphorylation and acetylation controlling activity on the spot. The analogy of modification sites being like allosteric sites with attached ligands makes sense. And phosphorylation often hits early pathway steps under neural hormonal control.
That's a common pattern.
And this idea that acetylation levels might reflect energy status via acetyl-CoA and NAD plus NADH levels that directly links metabolism to enzyme regulation.
Yes, it shows how the cell's metabolic state can directly feed back into controlling its own machinery. A very tight loop.
Right. We've covered a lot. Homeostasis, passive-active flow control, enzyme quantity control via synthesis degradation, and rapid activity control via allosteric and covalent modifications. The final piece is how these all connect into sophisticated control networks.
That's the ultimate picture. These aren't isolated switches. They're nodes in complex, integrated information processing networks. That's how cells achieve coordinated, nuanced responses to countless internal and external factors.
And the eukaryotic cell cycle is the example given to illustrate this network complexity.
It's a perfect example. The cycle G1 growth, S-phase DNA replication, G2 prep, M-phase division is tightly controlled by networks involving checkpoints.
Checkpoints to ensure one phase finishes properly before the next starts.
Exactly. Figure 9 to 8 shows a simplified G1 to S checkpoint. If DNA is damaged, a sensor kinase, ATM, gets activated.
Okay.
ATM starts a phosphorylation cascade, activating other kinases like CHK12. These then phosphorylate and inhibit a phosphatase called CDC25.
And what does CDC25 normally do?
It normally activates the CDK cycling complex needed to start S-phase DNA replication. But when DNA is damaged, CDC25 is inhibited, the CDK cyclin stays off, and the cell cycle arrests in G1.
Preventing replication of damaged DNA.
Precisely. It buys time for repair. And if these checkpoints fail, as the source notes, mutations can accumulate, potentially leading to cancer. It shows the critical role of these networks in maintaining stability. And that's just one checkpoint. The whole cycle is wired with these complex signaling loops.
A really clear example of kinases, phosphatases, feedback, all working together for precise control. So, wrapping up this deep dive, we've journeyed from homeostasis down to the molecular nuts and bolts of regulation.
Passive versus active flow, enzyme quantity, catalytic activity.
Allosteric effectors, feedback, indicators, second messengers, covalent modifications, irreversible proteolysis, reversible phosphorylation, and acetylation.
And finally, seeing how they integrate into these sophisticated networks like the cell cycle control system.
It really is a testament to the incredible complexity and precision that allows life to adapt and function.
The elegance of biological regulation is truly remarkable.
It definitely leaves you thinking. We touched on a few diseases linked to regulatory failures, but just imagine how many other conditions might involve subtle or major disruptions in these fundamental pathways, and what new therapies might emerge from understanding how to target these networks, how to nudge them back into balance.
It's a huge frontier for research, absolutely. Understanding these principles is key to tackling so many health challenges.
A fascinating area indeed. Well, thank you for joining us on this exploration of the intricate world of metabolic regulation.